and you can begin whenever. We've been at 26 participants for a couple minutes, or for a minute.
All right, perfect. Sounds good. So my name is Allison Grenell. I'm a PhD candidate in the Department of Pharmacology at Case Western Reserve University, and I do my research with Dr. Bela Anand-Apte at the Cleveland Clinic Cole Eye Institute. And today I'm excited to talk to you about my work exploring the role of extracellular matrix on retinal pigment epithelial cell metabolism. Sorry, it wasn't going. All right, so just quick context for this work. So I'm focusing on retinal degeneration, which is the progressive loss of vision, which can result in complete blindness. And the most common form of retinal degeneration is age-related macular degeneration. And I've included a photo here of my cat Finnegan to illustrate what your vision might look like if you had age-related macular degeneration. And while we know that the vision is lost because photoreceptors die, we don't quite understand what is actually triggering this cell death. So the aim of my research is to try to understand the pathological mechanisms underlying photoreceptor cell death. As everyone here is well aware, the retina is one of the most metabolically active tissues in the body, and to meet their high energy demands, photoreceptors utilize a process called aerobic glycolysis, where glucose is converted to pyruvate and then lactate, and this is in contrast to the pyruvate going on to enter the mitochondria and the TCA cycle and undergoing oxidation to generate ATP. So with the retina relying on aerobic glycolysis so much, it needs large amounts of glucose to be delivered to it. And it's the job of the RPE to provide the photoreceptors with this glucose. And while the photoreceptors are more of a picky eater, preferring glucose, the RPE cells are kind of the opposite of a picky eater. And they utilize a variety of different substrates, kind of acting as scavengers to utilize everything they can except glucose, so that glucose can be conserved for the photoreceptors. So some examples of those substrates are fatty acids, glutamine, and proline. Within the past decade, RPE metabolic dysfunction has been implicated in multiple different forms of macular degeneration, and it's believed that one of the reasons why this is so detrimental to the photoreceptors is because when RPE metabolism is altered, then they start to utilize glucose, which means that less glucose goes to the photoreceptors, and then the photoreceptors can dysfunction and starve. And while we really see the end product of retinal degeneration in the retina, many people believe that the root actually lies within the retinal pigment epithelium, or the RPE. The RPE is a monolayer of cells that lie between the photoreceptors and the choroidal vasculature, and they're responsible for a lot of functions that are essential for photoreceptor survival. And therefore, a lot of work has gone into the relationship, understanding the relationship between RPE and photoreceptors. But between the choroidal vasculature and the RPE cells is this extracellular matrix structure referred to as Brooks membrane. And this is a rather unique ECM structure compared to others in the body. It's a bit thicker than most ECM structures, and it also is striated, having layers of different extracellular matrix ligands. And the main function of Brooks membrane is to provide structure to the RPE and the retina, while also being permeable enough for nutrients to pass through it. And it also turns out that really early on in retinal degeneration, Bruch's membrane undergoes significant changes. And alterations to Bruch's membrane are one of the first pathological features that we can see if we look at post-mortem degenerating eyes. So here I've included two examples showing some of the changes that Bruch's membrane can undergo during aging and disease. So on the left, we have an example of Bruch's membrane during aging. So here's a 4-year-old Bruch's membrane where this line is representing where Bruch's membrane is, and here's an 81-year-old Bruch's membrane. And there is an increase in the thickness, as well as a decrease in the density with an accumulation of lipids and proteins. And it's also been found that with age, the membrane gets more stiff over time. On the right, I have an example of a Bruch's membrane during Sorsby's fundus dystrophy, which is a monogenetic form of retinal dystrophy similar to AMD. So we have the healthy Bruch's membrane up here and SFD Bruch's membrane, where again, we have an increase in the thickness as well as an accumulation of proteins and lipids within and around the extracellular matrix. Bruch's membrane also extensively changes during age-related macular degeneration, and this has been fairly well characterized. So this is just an image showing some of the things that we know occurs during AMD within Bruch's membrane. So there's a change overall in the composition of the membrane. There's an increase in profibrotic proteins such as collagen and a decrease in other proteins such as laminin. Again, there's an accumulation of lipids and proteins around and within the membrane, as well as an increase in thickness and the rigidity or the stiffness of the extracellular matrix. So while the field is becoming fairly confident that RPE metabolic dysfunction is driving photoreceptors to die and vision to be lost, the cause of the metabolic dysfunction has yet to be determined. And since the turn of the century, it's been continuously reported that the first pathological changes that we see are within the RPE extracellular matrix. And this led me to wonder if the ECM disruption could be contributing to the metabolic dysfunction within the eye. So I hypothesized that alterations in Bruch's membrane are contributing to RPE metabolic dysregulation. And to study this, I decided to look at two different models of Bruch's membrane dysfunction. The first being a genetic model of Bruch's membrane dysfunction, which is Sorsby's fundus dystrophy, and the second being a biophysical model of Bruch's membrane using synthetic hydrogels. So I'm going to start out talking about our findings in our SFD model. So as mentioned, SFD is a monogenetic form of macular degeneration that manifests similarly to AMD within the eye. And it is caused by mutations in a protein called TIMP3, or tissue inhibitor of metalloproteinase 3. This is a protein that is in the extracellular matrix, and there it specifically inhibits matrix metalloproteinases, or MMPs, which are proteins that cleave ECM ligands. So this makes TIM3 important for maintaining extracellular matrix dynamics and homeostasis. So we started this project using a broad approach, and we did proteomics and RNA-seq on our SFD mouse. So this is a mouse that expresses the S179C variant of TIM3, and we did proteomics and RNA-seq and then analysis using Panther gene ontology. These graphs here are representing the protein class and cellular processes found to be associated with the proteins that were differentially expressed between our mutant and wild type SFD ICUP. And when we did gene ontology analysis, we found that many of these proteins were associated with metabolite interconverting enzyme and cellular metabolic process. We also conducted RNA-seq, and similarly, we found that these were the two most enriched protein class and cellular process associated with the dysregulated proteins. So this was really our first clue that there was something going on with metabolism within the SFD mouse. I also combined the proteomics and the RNA-seq data to do a Panther pathway analysis to see if I could identify any specific metabolic pathways that were dysregulated. And this right here is a graph showing all of those pathways that were enriched among the dysregulated proteins and transcripts. And the only overt metabolic pathway that we were able to identify was glycolysis. And interestingly, this only came up in our proteomics data. And we think that this might be because these enzymes are generally regulated at the protein level with post-translational modifications. So in order to directly study glycolysis, I turned to a TIM3 cell culture model using ARPE19 cells. So these are immortalized RPE cells that are expressing the S179C variant of TIM3. And I let these cells fully differentiate and then conducted uniformly labeled glucose isotopic tracing for 24 hours so that steady state was reached. And then first just looked at pyruvate and lactate to try to get a sense of the glycolytic activity within these cells. So first, just looking at the abundance of intracellular pyruvate and lactate, I found that there was an increase in both. And then when I looked at fractional enrichment, I found that there was an increase in labeled pyruvate enrichment, although there was no change in lactate enrichment. So together, this suggests that there is an increase in glycolytic activity. I also looked at, after 24 hours, the extracellular metabolites that were still in the media, and specifically at extracellular pyruvate and lactate, which were both increased, suggesting increased production of these, and then extracellular glucose, which was decreased, suggesting increased utilization. So together, this suggests that there's increased glycolytic activity. But ARPE19 cells are not the best model to study metabolism, especially since they originate from cancer cells. So I wanted to move over to a more physiologically relevant model. So with the help of Dr. Ruchira Singh, I was able to obtain SFD iPSC cells. And these were from patients that had the S204C TIM3 mutation. And so these were skin cells that were collected, dedifferentiated into iPSC cells, and then differentiated into iRPE cells. And as a control, CRISPR was used to correct the point mutation within these cells. And then I passaged them and expanded them, let them grow until they were fully differentiated, and then looked at extracellular and intracellular metabolites. The first thing I did was verify that there was also an increase in glycolytic activity like we saw in the other ones, and I'm not going to show you that data just based on time. But the next thing I did was just overall look intracellularly and extracellularly at what metabolites were altered. So when I looked extracellularly, I found that the mutants had a decrease in extracellular glutamine after 24 hours and an increase in extracellular glutamate, suggesting an increase in glutamine metabolism or uptake. And when I looked intracellularly, I found that there was an accumulation of intracellular glutamine in the mutant, and a slight decrease in the intracellular glutamate. So from here, I really wanted to look directly at what was occurring with glutamine metabolism in these cells. So to do this, I used uniformly labeled glutamine. So this is glutamine where all of its carbons, all 5 of them, have been replaced by heavy carbon 13C. And then we can look at the amount of labeled carbons incorporated into downstream metabolites in order to understand how much glutamine is feeding into the TCA cycle to replenish intermediates. So when I did this, I actually found, surprisingly, that there was no change in metabolites generated via oxidation or reductive carboxylation of alpha-ketoglutarate, suggesting that there's no change in glutamine contribution to the TCA cycle. Interestingly, though, I did find that there was a difference in M3 pyruvate and M3 lactate. And this can be generated when glutamine enters the TCA cycle and then is acted upon by malic enzyme, which produces M3 pyruvate. So this decrease in M3 pyruvate and lactate suggests that there is a decrease in malic enzyme activity. So that is basically the entirety of my findings in the SFD, regarding SFD metabolism. And I found that protein and transcripts associated with metabolism were dysregulated. And then when I looked into metabolism directly, I found increased glycolytic activity and glutamine uptake with no change in glutamine anaplerosis, although I did find decreased malic enzyme activity. So overall, this suggests that mutations to Brooks membrane proteins results in dysregulation of glucose and glutamine metabolism. Are there any questions before I go on to the next part?
I have something that's between a question and a comment.
Okay.
So if you see less pyruvate that gets labeled from glutamine, less M3 pyruvate that gets labeled from glutamine, that was fractional labeling, right?
Yes.
So to say that it's less, and pyruvate pools are increasing, is that right? Intracellular?
Pyruvate pool, yes.
So could it be that actual total levels of malic enzyme activity are the same, but the relative to other metabolic processes, malic enzyme activity is decreased. You know what I mean? Because when you're doing these
Yes.
steady state experiments, it's like all relative to every other metabolic process that's happening.
Yes. That is a good point. And I can definitely think about that more and try to think of maybe another way I could like directly look at malic enzyme activity. Do you think that would be a good idea?
Yeah, I mean, if you do a kinetic experiment, you could probably make a clear statement and not looking at, percent labeling, but looking at like change in moles of M3 pyruvate over, 0 to 4 hours, something like that.
Yeah.
Or, another sort of even more simple approach would be isolate the cells and do something totally like enzymatic assay type of thing.
Mm-hmm. That's what I was thinking, something along the lines of that.
It might require less material to do it that way.
Yeah. I'll keep thinking about that. Are there any other questions?
Charan Deep wants to add to the answer.
So I think I don't know if you can hear me. There's some issue with my camera. I think my camera doesn't like me anymore, but that's fine. So I think, I mean, if the pyruvate levels, total levels, and also the labeling is high, if we have to calculate mole percent, like how much increases there, probably that's going to be higher in the, I mean, altogether it's cumulative. So it's going to be a lot more. So if the pool sizes were opposite, then I think, yes, there is a issue with the labeling. But if with 24 hours and a steady state, so I guess like it's a higher activity of malic enzyme. Both of the data imply the same.
Well, the pool size is up and the labeling is down. So when you just account for, like, I don't think you can necessarily say that sort of total enzyme activity is up because you don't know whether or not the labeling is down due to an increase in pool size or less activity.
Yes, but also we have to consider pyruvate carboxylase backflux because that also enters into TCA, and the shuffling is like too fast to like answer that. Probably a time course is better in that sense, to have like a time course for like the mutant and the wild type with the glutamine, and look over labeling over time, and probably that may answer.
So I got a question. So the with the TIMP3 mutation, does that mean that there's altered activities of the matrix metalloproteinases? And if that's true, could there be differences in the pool sizes of amino acids in the medium because the enzyme, the protease is more or less active? I'm not sure which, but could alterations in the total amount of amino acids be affecting the fractional labeling?
I think yes, because of proline, maybe because proline is released from the matrix, and that
Collagen.
may go into glutamine. Yeah, from the collagen, yes.
Yeah. And proline feeds into alpha-ketoglutarate.
Glutamine. Yes,
Yeah.
exactly. Yeah.
Yeah.
Correct.
I think that it seems that there are mixed publications out there whether of how MMP activity is changed with mutant TIMP3.
Yeah.
I've seen some reports of increased, some reports of decreased activity. But there are differences in intracellular amino acid levels, I believe.
In your work, is there a difference in extracellular amino acids? Or I guess you've already
Yes.
shown us glutamine and glutamine.
Yes, there is a change in extracellular amino acids. There is an increased utilization of phenylalanine, serine, at least 3 other amino acids in the media. All right. So next, I'm going to talk about the biophysical model of Brooks membrane that we've looked at using synthetic hydrogels. And the initial result that inspired the project to go in this direction was atomic force microscopy that was conducted in our SFD mouse. So here we used AFM, which essentially uses a cantilever to press down on the tissue and then measure the amount of displacement that occurs when you're pushing down in order to measure substrate stiffness and biomechanical properties. So we looked in 3 different regions of the eye cup, the central, intermediate, and periphery, and looked at the stiffness. So here, the 3 light colors are representing the wild type stiffness in the 3 different regions, and the dark color is the mutant stiffness in the 3 different regions. And we found that the wild type eye cups had a stiffness of around 20 to 40 kilopascals, where the SFD eye cups had around a 25-fold decrease in that stiffness. And there are many reports in cancer that changes to stiffness can impact metabolism significantly. So this got me really thinking more about metabolism and the biomechanical properties. So I used to study plant pathology, and I think a good analogy can be drawn between RPE and plants to kind of help illustrate my question. So if photoreceptors were trees, then RPE cells would be the roots that transport nutrients to the trees, and Brooks membrane would be the soil that they're rooted into. So then what I'm trying to understand is, if we were to have soft or hard soil, how would that change the nutrients that are actually getting to the tree? So to address this, I used primary porcine RPE cells, and I plated them on various synthetic hydrogels of different stiffnesses. So I've done this using two different substrates, polydimethylsiloxane and silicon, but I'm going to focus on talking about my results in the polydimethylsiloxane. So I used 3 different stiffnesses. The most soft stiffness is 2 kilopascals, and this is corresponding to the softness that we found in our mutant eye cup. Our intermediate stiffness is 16 kilopascals, which is closest to the physiological conditions that we found. And then the most rigid or stiff substrate is 64 kilopascals. And I wanted to include this because some have found that in AMD, there's actually an increase in the stiffness. And in order to get these cells to stick to the substrate, I tried multiple different ECM coatings. I ended up using polyl-lysine and laminin. And then I just started out by growing these cells and then observing them by light microscopy. So here are images of the cells on the substrate. So we have the softest to the most rigid over here at day 3, day 5, and day 9. And I found that at day 3, there was less cells on the 2 kilopascals, which corresponds to either a decrease in attachment or growth. And then at day 5, the cells on the softest never really formed complete intact monolayers. So if you look closely around where my pointer is, there's still some areas where cells have not completely closed in and filled those gaps. And then these holes just expand and become bigger over time until there's large holes in the monolayer. And so I wanted to first understand if this was a difference in attachment or growth. So I assessed attachment by growing cells for 24 hours and then just using media to gently take off any loose cells, and then counted the cells that were still there. And I found that there was no change in the amount of cells, and that suggests that the difference that we're seeing at day 3 is probably because of a change in how the cells are growing rather than how they're attaching. The next thing I wanted to do was look at RPE cell markers to see if there was a change in maybe the differentiation state. So I looked at RPE 65 and NAK ATPase, and I found that there was no significant difference in these markers, which I did find to be a bit surprising because of the holes that did develop in the monolayer. I thought that there would be a change in these markers, but I did not see any. And then the last the last thing I did, actually looking at the cells, visualizing the cells, was looking at the blood retinal barrier, the tight junctions that form between the RPE cells. So here I have the different stiffnesses. This is staining done at day 5. And we have the bright field, and then Z01 and Claudin 19. And in the stiffest conditions, or in the softest conditions, we see that there is less CO1 and Claudin 19. That increases a bit in 16 kilopascals, and then there is the most tight junctions in 64. So this suggests that the substrate stiffness can actually control the blood retinal barrier. So then I wanted to specifically look at metabolism. And so I started off by just, again, looking at the amount of metabolites intracellularly and extracellularly. And the metabolite that I found to be the most altered was intracellularly was glutamine. And there is less glutamine intracellularly in the cells on the softest substrate. And when I looked extracellularly, I found that after 24 hours, there was less glutamine in the media for the softest substrates and more glutamate in the media on the softest substrates, suggesting an increase in glutamine utilization. So although there were low intracellular glutamine levels on the softest substrate, there does appear to be an increase in the amount of glutamine that's being taken up. So I wanted to look directly at glutamine metabolism first to see what was going on. And this is showing us the fractional enrichment, first from glutamate and proline, which are generated from glutamine. And I found that on the softest stiffness, there was less glutamine and less proline, signifying a decrease in the contribution of glutamine to these metabolites. And when I looked at TCA cycle metabolites, ones that could be generated from both oxidation and reductive carboxylation, I found that there was, again, a decrease in the fractional enrichment of these metabolites from glutamine, suggesting a decrease in glutamine contribution to the TCA cycle and anaplerosis. I then wanted to also look at glucose to see if glucose was changing. I hypothesized that if I was seeing this large change in glutamine metabolism, then I would have seen like a compensation in glucose. So just starting looking at fractional enrichment of labeled pyruvate and lactate generated from uniformly labeled glucose, I found that there was an increase in enrichment in the softest substrate, but not by as much as I would have expected. And then I also looked at the contribution to the TCA cycle, and I got some puzzling results in my enrichment. So to try to make sense of this, I decided to kind of break it down to try to understand the contribution coming from pyruvate dehydrogenase versus pyruvate carboxylase, and then total glucose contribution to the TCA cycle. So to look at pyruvate dehydrogenase, I looked at the ratio of M2 citrate to M3 pyruvate. And I found that within the softest, there was a decrease in this ratio suggesting that the contribution through pyruvate decarboxylase in the softest conditioned is decreased. But that is not the only way that pyruvate can enter the TCA cycle. It can also enter through pyruvate carboxylase. So in order to understand the activity there, I looked at M3 aspartate over M3 pyruvate. And I saw that in the softest condition, there was a slight increase, suggesting there is an increase in the contribution via pyruvate carboxylase. And then to try to just understand the total contribution to the TCA cycle, I just looked at the amount of labeled citrate that was present. And I didn't find that there was any difference here. So this suggests that there's no change in the overall glucose contribution to the TCA cycle, although there is a change in the route at which the glucose is entering the TCA cycle.
I have a question.
Yes.
So looking at these ratios, and I guess I probably shouldn't be thinking about it this way, but if the ratio of M3 aspartate over M3 pyruvate is like 0.5, then does that mean that half of aspartate comes from labeled pyruvate?
Does that mean that half of aspartate comes?
Just trying to interpret the ratio in a way that I can derive. I mean, I think the point you're making is right.
I think that is correct, what you're saying. Yeah, like 50%.
That's a lot. That's, like a pretty high amount of TCA cycle carbons would then come from carboxylation, pyruvate carboxylation.
Yeah, that's true.
And I don't really know what the meaning of that is, but it's true in all your conditions. So I don't know. It's interesting. I have no other comment than that. It's interesting, I guess. But I mean, I thought I was going to try to make a comparison to carbons coming from carboxylation versus carbons coming from sort of normal pyruvate dehydrogenase activity. And it would seem that more come from carboxylation, but I'm not sure I can really make that comparison directly.
I think that's why we have to use bicarbonate buffers.
Jim, I'm not sure I'm not sure I followed what. Could you explain that more?
Well, I mean, to do carboxylation, you have to have CO2.
Oh, I see.
You have to have bicarbonate.
Yeah. Okay.
Otherwise, that reaction just won't happen. And
you mean
that's what
opposed to using a Hebe's buffer, you're saying, for example?
Yeah, right. Yeah. And I mean, that's always been to us one of the problems with seahorse. I don't know how significant it is, but they don't use bicarbonate buffers. So if these really are major reactions happening in these cells, then those aren't happening if you're just using Hebe's buffer without bicarbonate. So
I think likewise, you could argue that the concentration of CO2 might not be high enough for as much carboxylation to happen in my eye.
Well, you mean how much CO2 is present in our tissue?
Well, yeah, and whether or not that's whether or not the concentration is a main driver, or concentration of CO2 is a driver for carboxylation.
I have a question actually on this. So because all these experiments are done in 5% CO2 incubator and with the bicarbonate in the media, so will that still allow for the normal carboxylation to happen? Because there's bicarbonate, there's CO2, 5% CO2 in the incubator. So will we still need excessive bicarbonate?
I think what Jim's point is that there some people will do cell culture in totally bicarb-free conditions. They'll use heat.
Oh yeah, no, that's not good.
And because then if you take heapies out of the incubator, you'll maintain the pH.
Yes.
It also has its advantages for cell culture, but I don't want to speak for Jim. I think he was saying,
Yeah.
but for metabolism purposes, you need a source of CO2, and
Definitely. Now I got it. So yeah, okay.
Yeah.
Yeah, thank you.
I mean, the 5% CO2 is just to maintain a constant concentration.
pH.
Because
Yeah.
as soon as you take it out, like Jason was saying, the pH starts changing because the CO2 goes off into the atmosphere.
Yeah,
So,
exactly.
yeah.
Yeah, got it. Thank you.
Allison, could I ask what the total fractional labeling of citrate was with the M1 through M6 isotope logs, if you remember that?
The total fractional labeling. Does this help?
Yes. So it's like 85 to 80-ish%. Okay. I'm just wondering if everything is just so well enriched that you don't have a large dynamic range to see changes that might exist. You know what I mean?
Everything is so enriched. No, I don't know what you mean.
You may want to have earlier time point in addition to this.
Like,
Because
sorry, go ahead.
I think you're saying like maybe the M3 is like so much that you cannot make further M3 of pyruvate and these compounds, right? Or the pool sizes are like already saturated to the level where you cannot exceed further.
Yeah, I like, if you're 100% labeled and
Yeah.
in that condition, if metabolism goes faster, you can't see it because you're 100% labeled.
Yes. Yes.
And you're not 100% labeled, but you're like close. And
Yeah.
so I'm wondering if you were, 50% labeled at, say, like what Charandeep said in an earlier time point, if you might have more flexibility to see increases in metabolic rate.
Mm-hmm. Well, I've done this at an earlier time point.
Is that another back pocket slide?
No, it's not, because my mass spec isn't working well enough to analyze the samples. But hopefully that will give us some more information here. I do have media samples over time too. All right. So to summarize, what we found in the second part, I found that RPE cells on soft substrates have blood-retinal barrier breakdown as well as altered metabolism. And like our SFD model, I found that there was increased glycolytic activity and increased glutamine uptake. But unlike our SFD model, I also found that there was decreased glutamine contribution to the TCA cycle with increased pyruvate anaplerosis and no change in total glucose contribution to the TCA cycle. So together, this suggests that ECM biomechanical properties can influence RPE, glucose, and glutamine metabolism. And this does support my hypothesis that alterations of Brooks membrane are contributing to RPE metabolic dysregulation. And I think that this suggests that the Brooks membrane integrity could be driving RPE metabolic dysfunction, and it warrants further investigation in the future. One thing that, so here's kind of a model or a summary of the results I found on the soft extracellular matrix. So increase in glycolytic activity represented here with increased intracellular lactate, and then decreased extracellular glutamine along with decreased intracellular glutamine. And and then decreased contribution to the TCA cycle from glutamine. So one thing that I've just been thinking over and over about is, where is this glutamine going? It's not going to proline. It's not going to the TCA cycle. Where is it going? And I've been throwing around two ideas. My first idea is it even getting into the cell? because because even when we look at intracellular glutamate enrichment, that's already it's decreased, it's already decreased there. So I'm wondering if there's just somehow the glutamine is being metabolized extracellularly to glutamate, and then that glutamine isn't even getting out of the cell, or it's going in really quick and getting exported really quick. So I've been trying to think about that. The other thing I've been wondering is if it could possibly be being used by the hexosamine pathway. And so the hexosamine pathway utilizes both glucose and glutamine in order to generate hyaluronan and protein glycosylation. So here's just a graphic of how that is. So we have glycolysis right here, and the hexosamine pathway branches off from glycolysis, and glutamine, the rate limiting step of the hexosamine pathway is GFAT, where glutamine is converted to glutamate. And we did find that in our SFD model that there is an increase in hyaluronan deposition in our mutant TIM3. These were from our ARPE19 cells. So that is something I'm also wondering is, could this possibly be going, using the glucose and the glutamine in order to generate hyaluronan? And hopefully I'm going to test that as soon as my hyaluronan ELISA kit comes in. And that is the end of my presentation. I would like to acknowledge that this work has not been done alone. I have gotten a ton of help from everyone in my lab and a lot of outside advice, especially Dr. Jenhai Du, Dan Haas, and Charan Sign. I've asked them so many questions and gotten their help so many times. And with that, I will take any questions.
Allison, this is Jason Miller. I'm at University of Michigan. Is there, just to answer the very basic question of where glutamine's going, I had forgotten or was unaware of glutamine's contribution to the proteoglycan pathway, like you said. But couldn't you just harvest your cells at various time points after you introduce the extracellular glutamine and just measure how much C13 glutamine you have, in the cell at various time points? That would give you at least a consumption rate, because of course one also alternative is that it's being incorporated into proteins or other things where it's not being, we're so metabolism focused that we forget that amino acids, primary purpose is to build proteins. So like, couldn't you sort of pick that apart a little bit by tracking how much gets into the cell and what its consumption rate is?
Do you think intracellular glutamine would be the best way, or would looking at consumption of extracellular glutamine?
Well, you already showed
Yeah.
that extracellular glutamine is going down.
Yeah.
So you want to know whether or not intracellularly, whether it's hanging around as C13 glutamine or being quickly converted into something else. Right now, you don't know that, right? Because you're not measuring C13 labeled glutamine intracellularly, you're just measuring total glutamine intracellularly.
Yes, I have.
If you track the label itself, you'll know the consumption rate, and then that at least tells you how actively whatever process it's going to, it'll tell you how actively it's being utilized.
So I have looked at actual C13 glutamine,
Okay.
Intracellularly. And I can't remember right now what it was for the different stiffnesses, but I know that in my TIMP3 model, it's unchanged. But I can't remember, but I will definitely look into that.
Unchanged meaning not being consumed, or the consumption rate's the same as
Consumption, at least the intracellular amount of labeled glutamine was the same
across the conditions.
across the conditions.
So to support the protein synthesis notion, I think we've talked about this briefly perhaps, but if you isolate protein and then hydrolyze it to individual amino acids and measure, say, fractional labeling on those amino acids, you might see if that is changed. And I think the proteomics people at Cole would probably know how to do that. That's like when I was talking about the protein concentration estimation, since they're familiar with that protocol, it might help. And then alternately, glutamine utilization, I'm sure I've talked about this, glutamine utilization, if it's up and glucose utilization is just up more, and you're not really seeing the increase in glucose utilization well because you're super well labeled, then it could still explain all the data. Like there's just drastically more metabolism that's happening, and it's more, I mean, I guess, I forget, did you show glucose consumption as a function of stiffness?
No, I did not.
Is that increased with higher stiffness?
I have not looked at that yet.
Okay. That might be interesting. It would give you a sense of whether or not there is a sort of, at the same time as more glutamine consumption, there's also more glucose consumption.
Yes. Yeah, no, I think that is really important, and which is I'm going to use your glucose enzymatic assay that you shared the protocol to try to figure that out.
It's not mine. It's, there's a book that I used, but thank you. Yeah, I'll.
Well, you wrote it up very beautifully.
At Jason's request, actually.
Really?
Yeah.
Yeah, I said it was too valuable not to have it written up. I mean, this just what Dan's saying, just this idea of, if everything is revved up, just knowing the key players that you're looking at and their overall, consumption rates, either by tracking, I mean, again, you could do it by tracking the C13 label if you're not, if you want to be sure that it's not coming from something else, like, proteolysis of pre-existing proteins or whatever else. But then you would really know, like, just how, the key metabolites, how much are they being consumed? And if they're all being consumed faster, then the balance between them is not going to be different.
Allison, here's Claudia from UMass. I had a question. I mean, you said it, but I don't remember. Why did you focus on glycolysis and not on the Wnt pathway, where you had a lot of hits, which is known to regulate the extracellular matrix and important for these kind of processes that you were studying?
So why did I focus on metabolism over? Although Wnt can control metabolism, but I just figured that, glycolysis, that was the main pathway I got, or the only pathway I got. So just start with that to directly evaluate metabolism. And then I just haven't had time to go further into the Wnt story.
Okay. Yeah, I mean, it's very possible that you just have a compensatory mechanism, right? From RP cells, they're shifting around amino acid synthesis and things like that to make up for the loss in the extracellular matrix, right? And that's why you're seeing these shifts, and they're trying to synthesize some other extracellular matrix proteins. And it's a compensatory response, right? And in your other model where they don't stick well, they are unhappy and try also to secrete maybe stuff that helps them to stick better. or construct something more rigid, right?
Mm-hmm.
And that may be the reason you're seeing the response you're seeing.
So you're saying that the proteins, so it's making extracellular matrix ligands.
Yeah, ligands or proteins that are part
Yeah.
of the extracellular matrix. Like, I mean, you see change in hyaluronic acid, right? If you look at other known extracellular matrix proteins of the Bruch's membrane, right, are they trying to compensate? Like, if with TIM3, if you preferentially degrade a certain protein, are they trying to compensate with synthesizing different proteins that help them stick better, right? And you're seeing a metabolic shift, but it's because the cell is trying to synthesize more of one thing and less of something else, just to kind of stick better to the surface.
Yeah, that's definitely a possibility.
That's a neat story. If it was true that there was like a synthetic shift corresponding to the different matrix, and then it would also be neat to know if the shift in synthesis, I mean, not to just take everything back to energy metabolism when it's not necessarily like the main thing, but if the shift in synthesis did sort of either increase energy demands to an extent that was unhealthy to the cell and sort of starved it, yeah, I don't know. I think regardless, looking at protein synthesis rates might be really critical. I agree with Claudio.
Oh, hi, Allison. I have a few questions for you. Have you looked at the RP flat mounts of the TIP3 mutant mouse?
Yes. Well, not me particularly, but many other people in my lab.
Because it seems like based off of your experiment with the stiffness of the of Brooks, are there changes happening with the RP morphology or RP number?
Not really. The changes are pretty minimum that we see in the mouse.
Okay. Which that kind of leads into my next question. For clarification, the TIP3 mutant mouse, that mouse is characterized by a lot of angiogenesis in the retina, correct?
Theoretically,
Because
I don't, but just overall, honestly, in our mouse, we do not see very many changes. Like flat mounts, like most of the changes that we see would be like if we were to introduce sodium iodate in, or if we were to injure with a laser, like that's when we see the most pronounced changes. But without like some sort of stressor at baseline, there's really not very many changes, which was another reason why I was trying to steer away out of the mouse model and, try to find some other ways to look at this.
yeah, because it got me thinking about what you're finding, and it kind of goes back to the question. It's like we see all these changes and how much of it is contributing to the pathologies. That one thing that it's really interesting with the stiffness changes that you're observing, that I would also assume that that would be affecting your choriocapillaris, that if that's becoming much more loose, it's like, could that lead to the growth of blood vessels into your RP and photoreceptors?
Mm-hmm.
But, yeah, good talk. Thank you so much.
Thank you.
So I just asked this in the chat, but I'll say it. How long does it take for TIM3 to cause pathology in people? I think you might have maybe even mentioned this.
It's typically like after 30 years old.
Okay.
30, 40 years old.
Allison, do you know, I mean, the second part with doing the stiffness was cool. Do we know what the stiffness, maybe you said it, but do we know what the actual stiffness of the wild type versus mutant mouse Brooks is? Like, have those experiments been done testing stiffness, some sort of AFM based stiffness testing?
Yeah.
I'm sorry, I missed that.
It's okay. This
That-
was our AFM results
So
in
they're
the,
also, it's all softer,
yes, it's all softer, but this is in the mouse.
and we don't know in humans, like that's, you know,
No, we don't know in humans.
So we should take 64 to be closest to your control. Is that right? In your
I was saying more so like 16 closest to the control.
Yeah, like peripheral.
64 is relatively stiff in terms of like tissues.
one thing, I mean, I'm not inventing experiments for you, but if you, has anyone denuded Brooks on on these Sorsby's versus wild type mice and then tried to do RPE, introduce RPE culture on them? You know, obviously it was really cool to have this in vitro model of changing the stiffness by changing the synthetic properties. But
Mhm,
of course the actual Brooks and, you know,
mhm.
is most relevant is the actual what's most relevant is the actual Brooks, which has both signaling and stiffness properties to it. And obviously you'd have to count on mouse human signaling being conserved because it's a mouse Brooks membrane. I feel like I've seen some people try to do that type of thing. Be curious if the RPE grows differently on the Sorsby's Brooks compared to the non-Sorsby's Brooks.
So I haven't seen it with Sorsby's, but I know that people have done it with human AMD.
Mhm.
And I don't necessarily remember all of the changes, but I know that there are RPE changes that happen when you plate RPE cells on AMD Brooks.
Right. The problem is there's lots of, like, I think a lot of those experiments also were like, oh, well, Brooks has been oxidatively modified. And so it's a lot, there's a million things that could happen. Whereas in these mice, they don't have much of a phenotype. So you then presume that that there's not that much toxic signaling, at least going from the Sorsby's, but you clearly show differences in stiffness and you, from your guys' work previously, show compositional differences and structural differences. So maybe you would more isolate those effects of a Sorsby and their effect on the RPE
Mm-hmm.
than you would with like some AMD Brooks eyes.
Mm-hmm. I do think logistically it would be pretty hard.
Yeah. So you can just cover your ears and say, and say, that's nice. I won't do that.
Yeah, I think it's nearly impossible to isolate Brooks from a mouse. It's just too tiny.
Yeah, it's so small.
Yeah.
And another thing is, right, like these results are showing that the stiffness is also changed spatially. Like it's more stiff centrally than peripherally. So I think that that would kind of make it even harder in a mouse because we wouldn't be able to isolate those specific regions.
Do you think the different stiffnesses in different regions would mean that RPE cells are going to have regionally different metabolism?
I think yes.
I feel like it's kind of tough. Like maybe we shouldn't be seeing big effects in a model of AMD or of SFD if the disease takes 30 or 60 or more years to develop. I mean, it's kind of, I guess, an ongoing discussion that everybody has, but if the disease takes that long, then why should it be immediate in culture?
Mm-hmm.
Because we're impatient.
Yeah. No, I mean, that's the only answer.
And there's only $2 to go around.
Yeah. Okay. Thank you so much, Allison. This was great.
Thanks, Allison.
Thanks, guys.